Mitosis. Mitosis is cell division to form two genetically identical cells. So before we start to look at the process, we need to look at the structure of the chromosome. The chromosomal structure is important in mitosis because it explains how it is possible to enable one cell to divide into two genetically identical cells. It all comes down to these things here, which are the chromatids. So one chromosome is made from two chromatids, and those two chromatids are completely genetically identical. So in mitosis, when the chromatids are separated, when replication occurs, you're able to make an exact copy and therefore make exact copies of the chromosomes. So, how does the cell cycle operate? Now bear in mind that this is a cycle and it has been divided into stages, but the stages do run into each other in a completely smooth pathway. And so, late prophase and early metaphase are kind of similar. We start off with interphase. Interphase is the longest period of the cell cycle and it is the non-dividing part of the cell cycle. The cell will spend the majority of its life in interphase, with only a small fraction of it being spent in the dividing phase. Interphase itself has three phases, G1, the S phase and G2, and it is in these stages that the DNA replicates in G1, that the organelles are synthesized in the S phase, and then in G2 the chromosomes start to condense. The first dividing stage is prophase, and in this stage the chromosomes condense, the nuclear membranes disintegrate, the spindle fibres start to form, so that means that the centrioles move to the poles of the cell, and the chromosomes will start to attach to the spindle by the centromere. Now if we look at this electron micrograph, you can see that in early prophase you can start to see chromosomes forming, whereas in late prophase you've got distinct chromosomes. Compare that to a cell over here, which is probably an interphase or perhaps very, very early prophase. The next stage is metaphase, and in metaphase the chromosomes align on the equator of the cell. You can see that quite clearly here, right in the middle of the cell, the chromosomes have all aligned, ready for the next stage. And that next stage is anaphase, and in anaphase the centromere splits and then the chromatids start to move towards the pole of the cell. As you can see here, the chromatids are being dragged along to the pole here and dragged along to the pole here. That then moves into telophase, and in telophase the chromatids are now at the poles and the nuclear envelope starts to reform around these two nuclei. Which just leaves us with cytokinesis, which is the final division of the cell to make two new cells. It is worth saying that cytokinesis in animal cells, which is just caused by the invagination of the cell membrane to sort of bud off the two new cells, and in plant cells where the vesicles containing the components for the cell wall align at the equator and then secrete the cell wall into the gap, and then the vesicles sort of join together to make the cell membrane. Those two processes are obviously quite different in the two different cell types. This is a great picture which shows the different stages of cell division as it would be on an electron micrograph, or even actually potentially a good light microscope. And you can see the different phases quite clearly. So metaphase is probably the one that's always the easiest to find, with the chromosomes being aligned at the equator. Anaphase is another one that's easy to see. There are lots of cells here in interphase showing maybe a little bit of chromosomal condensation, but basically just a senescent nucleus. And then prophase, far more definition. This would potentially be either very late prophase or early anaphase, and then telophase where you have the two cells. Okay, so being able to recognise the status of mitosis from electron micrograph drawings is very important.